Hi, everyone. Okay, I'll go ahead and get started. So I'm Hera Vlamakis. I'm a group leader in the microbiome program here at the Broad, working with Ramnik Xavier and Curtis Huttenhauer. And I'm just going to give you a big overview on microbiome research in general today, how we do things, make sure that we're all on the same page as far as some of the terms that we use, and then I'll give you a few short stories of work that we've been working on to show how we think the field is headed. Okay, so we have about as many microbial cells in our bodies as human cells. This says 38 trillion in my computer. And most of those bacteria are in our gastrointestinal tract, while our 30 trillion human genes have our 30 trillion human cells have about 20,000 genes that are encoded in them. In our microbial population, we have 8 million genes. So even though the cell numbers are about the same, the genetic potential of these microbes that are living in our bodies is humongous. And there's up to 10,000 different species that can live in the gastrointestinal tract. And most of these bacteria are good for us, so they do a lot for our health. They're essential. They help synthesize vitamins that we use. They help to take up different carbohydrates. Wow, this is totally messed up here. They help take up or break down carbohydrates from food that we eat, and they can prevent colonization by pathogens, as well as help to train our immune systems. And sometimes those microbes can be associated with disease. So in particular, one of the diseases I'll focus on today is inflammatory bowel disease, where we know that there are specific microbes that are enriched or depleted in this disease, and a dysbiosis of the gut microbial community plays a big role in that disease. And so, like I said, just to make sure that we're all on the same page as far as terminology, I'm going to go over a few of the words that we use commonly. So microbiome, metagenome, and microbiota. Initially, the word microbiome was used to describe the collective genome of all of the bacteria that live in your body. And I say bacteria, I should say all of the microorganisms. So these could be eukaryotes as well. And the term microflora or microbiota was used to describe the microbial community, so the actual bacteria. And more recently, the word metagenome has been used to describe the collective genomes of a community, so all of the DNA. And what I'll be using today is this. So I'll say microbiota for the community of bacteria. I'll use metagenome to describe the total DNA for a community, and I'll use the word microbiome to kind of encompass both of those. And so we have various big omics ways that we can do microbiome research. Basically, you start with your favorite sample. In the case of the gut, we use stool frequently. You extract DNA, then you can look at which bacteria are present using 16S ribosomal RNA gene sequencing. So this is an amplification technique. You amplify a specific gene I'll go into in a moment. It's great because it allows you to detect all of the community members, even the ones that are present at very low abundance. Alternatively, you can look at total DNA and do metagenomic sequencing or whole genome sequencing. This allows you to get all of the genes that are present. It doesn't just give you taxonomy, but you can actually start to look at function. And we'll talk about the pros and cons of these two methods. Also, we're looking at RNA to see what genes are actually transcribed. So just because you have genes doesn't mean that you're actually expressing them. And as we start to look at different data types, we can get a better idea of which bacteria are really contributing in a given environment at a given time. And finally, I should say that in order to study this community, we need to think about lots of things. I'm not going to go into too many details about other omic methods, but this is something that's ongoing in the field where we're looking at metabolomics, proteomics, viromics, culturomics, basically trying to get as big and broad a picture of the environment that the bacteria are living in. Okay, so let's go into a little bit of detail on 16S gene sequencing. This is the structural component of the prokaryotic ribosome. We use it to identify phylogeny. The reason why we can do that is because this gene is very highly conserved, even among all domains of life. So you can see in green here are colored the identical nucleotides, and Homo sapiens are at the top, yeast, corn, and then microbes of all sorts. And you can see that there are very conserved regions in the sequence. But the reason why this gene is useful is that those conserved regions span or flank very variable regions. So we can design PCR primers to amplify the variable regions, and then we can compare those variable regions to determine which microbes are present. This is great because you don't need to be able to culture the microbe. A lot of microorganisms are very hard to grow outside of their native environment. It's a little bit tricky because there's more than one copy of the 16S gene in most microbial genomes. And so if you don't take into account how many copies of your 16S gene do you have, you can be a little bit fooled by microbial abundances. So you need to do some computational analysis or normalization to be able to really determine abundance of a given species or a given taxa. And using just the 16S ribosomal RNA gene, you're actually a little bit limited in what level of taxonomic identification you can get. So it's very hard to get to actual species level identification. Most of the time you can only get to the genus level if you're lucky. And that's just because of the amount of sequence that we get. But when we have the sequence, you can look at the data in different ways. So here I'm showing you, these are all of the reads from a population, 16S genes that we have. Different colors denote different organisms. And so what we do is basically just count reads, and then you can group them based on organisms. So blue's got 5 reads, red has 7, et cetera, et cetera. We can determine the frequency, adjust for the copy number of the 16S gene, like I mentioned, and then you can visualize using a heat map for the abundances. And you can use, so I say organism here, but that's really operational taxonomic unit, or OTU. That's because frequently we can't determine a particular species. Like I said, we have many databases now of 16S ribosomal RNA genes, and we can compare our sequences to them to get the best or most similar taxonomic classification. So this is using 16S sequencing data. We can also classify microbes using the metagenomic profiles, so all of the DNA in that community. And so one tool that we use to do this is called MetaFlan2, which was developed with Curtis Huttenhauer's lab and Nicola Segata. The way this works is if you look at all of the organisms on a tree here, you can see that there are branches. We look for specific genes, so colored in red here. You could see this gene is only present on this clade. And so we're calling those marker genes because they tell us something about the phylogeny of the organism that they're coming from. And we know that if you have this gene, you're only present in this particular organism. This is based on reference genomes, right? So really, we rely on having a number of microbial reference genomes already sequenced, which there are many. So doing that, then we could look at those available reference genomes. We get our marker genes. We have a database of those genes. And then you can map your metagenomic reads to the database, and you can then assign taxonomy to those to the organisms that are present. And in the end, you end up with something very similar to what I showed you previously. You have, in this case, we can get to the species level because we're relying on much more sequence coverage to be able to classify things. You can see what species are present, and in each sample, so the columns here are samples. The species in those samples are in each row, and the boxes are colored by their abundance. And so you can get heat maps like this to visualize your data. Another way to look at whole metagenome sequencing data is using a reference-free method. So in this case, we're not using marker genes that are based on reference genomes, but rather we're using gene abundance. So we do the deep sequencing of all of the communities, and then you can take and make a non-redundant gene catalog. So you can cluster the genes based on their similarity, and then you can look and see in all of the samples that you have, what's the abundance of each of these genes. Based on the abundances, we can cluster the genes by co-abundance, and then you end up with these groups, which we're calling metagenomic species. And in each group, then you have all of the genes that are present at a given abundance. So that this is assuming that if your genes are on the same chromosome, you'll have the same number of each of those genes. They group together. And then you can use references to assign taxonomy. Again, we rely on taxonomy for that. But you can see how many of those clusters actually are not labeled by reference genomes. So here I'm showing you the number of species that are present in all of these different samples. Each row is a sample, and colored in black are all of the clusters where the taxonomy is not known. So really, there's a huge amount of unknown space still in microbiome research, because we rely on what's known in order to determine what's present. But no matter which way you get your taxonomic profile, you can then visualize it in different ways. So like I said, we can do heat maps where we count the reads, or we assign taxonomy in the different ways I said. For each sample, we can get abundances of a different OTU. And then we need to find ways to compare these. So we can look at heat maps, but another common way that we use to look at microbiome data is ordination plots. And so the way to think about these they're a way to show multidimensional data in two-dimensional space, right? So just like we can visualize the different views of this motorcycle here, you can do the same thing with a microbial profile. So you could imagine if you have 2 microbes only present in a sample, you can very easily just plot the abundance of microbe 1 to microbe 2 for each of the samples, and you can see how similar they are to each other. When you've got 3 or more microbes that are present in the community, you need to go a step further, and you can use different distance matrices to get a value for how similar are these samples to each other, and then you can plot those values here. And so you look at variation between samples, and the distance between the points on this principal coordinate plot is reflecting a function of their similarity to each other. So when you look at a PCOA plot, the closer the dots are to each other, the more similar those samples are, and each dot encompasses an entire microbial community. So just to give you an example of that, with the Human Microbiome Project, this was a study done or published in 2012, where we looked at healthy individuals, and we looked at, I say we, it wasn't me, but we looked at healthy individuals and the microbial communities on different parts of their bodies. And if you look at those communities, you can see that, again, each dot here is a community. The closer together the dots are to each other, the more similar they are. If we color by body site, you could see that there are location-specific microbes. So that means that the microbes that are present in my mouth or someone else's mouth are more similar to each other than on my skin or in my nose, etc. So depending on what side of the body you are, a microbe is, they're likely to be more similar to each other. Okay, and then there was a second phase of the Human Microbiome Project, which was more recently published. And while these data that I'm showing you here is based largely on 16S profiling, in the second phase, we generated a lot more metagenomic data. So this allows you to get more to the species level of which bacteria are present, and it also lets you get even deeper to the strain level. So you could look at single nucleotide polymorphisms to determine how those members of each species is varying. So looking now at 2,500 metagenomes from 7 different body sites, You can look at a principal coordinate plot and see that very similar to what we saw with 16S data, body sites group together, so the stool samples are all clustering here. These are all the oral samples. You could see they break down a little bit further with plaque or the buccal mucosa or tongue. But in general, body sites, again, will group with each other. What's different is that we can actually see the different species now contributing to these differences. You could see the stool samples have all these species up at the top, which are not very present in any of the other body sites, and similar for all the oral sites, have a cluster of species that are very low abundance in other sites. And so then looking at this, because we have different body sites, we can start to ask, like I said, strain level questions. And so there's another tool called StrainFlan. This is just one of many options that are available, where we looked at variation in strains. And so once again, using those marker genes that I mentioned previously that we use to determine a given species, we can look at those marker genes, we can map our reads to those markers, and then we can look at single nucleotide polymorphisms, right? So for every particular marker, each sample has a certain sequence for the reads that are mapping, and you could see that there are different variations that group together. And then you can visualize this either on a phylogenetic tree, or you can make a principal coordinate plot to basically look at how similar are the strains from a given species between different samples. And so we could plot then looking at the distance between strains within a given site. So how different are the strains in your mouth, for example, to each other compared to other body sites? And you could see that there are some organisms, like Streptococcus salivarius, where that fall right on this line. So that means that the strains that you find in the mouth are as similar to the strains that you would find on the skin. And you could see that here, each dot is a sample that has this species in it, and the samples are kind of all mixed together. And this is in contrast to, for example, Haemophilus parainfluenzae, where you could see that there are clusters that form when we look at each of the samples. And if we color by body site, you see that the clusters for the tongue dorsum are together, the clusters for the tonsils are together. So really, depending on where you are, even in the mouth, you have more phylogenetically similar strains, which I think is pretty cool that we were able to visualize that. Okay, so now I'm going to move a little bit away from all of the body sites that I've been talking about, and I'm going to focus in on the gut. So most of the bacteria, like I said previously, are in your gut, and depending on where you are in the GI tract, you have a different microbial load. The greatest load by far is in the colon, where you have 10 to the 9th to 10 to the 12th microbes present. The bacteria vary depending on where you are. And in most of the studies that we do, we use stool as a proxy for gut microbes. And the reason we do this is because of ease of sampling. It's non-invasive. You can get a stool sample from pretty much anybody. And you can sample over time. And I'll tell you a few stories in a minute where longitudinal sampling has made a big difference in our understanding of what's going on in a given system. Okay, so in the GI tract, this is now HMP1 data looking at the healthy volunteers. You could see the relative abundance of each of these genera is on the y-axis, and that every sample, every individual is a column here. It's ordered based on Bacteroides, which is a very common genera or genus in the gut. And you can see that there's a big variation in which bacteria are actually present in any healthy person's stool sample. But there's a balance there, and so no matter what your microbial community is, it's in some sort of balance with itself, and it reaches homeostasis, and for the most part, you're healthy. The problem comes when you have a dysbiosis. So there can be an expansion of some pathogen. You could have reduced diversity, for example, due to antibiotic treatment. Or you could, for some reason, lose beneficial microbes. And then the shift changes, and these are the instances where we think that the microbial community in your gut could affect your health. And there are certain diseases where the microbiome has been implicated in disease onset and pathology, and the one that I'll focus on today is inflammatory bowel disease. Crohn's disease is one of the major subsets of inflammatory bowel disease. One of the interesting things here in this graph that I like to point out is that Crohn's disease and many other immune disorders, the incidence has been increasing over time. And this increase in incidence is likely due to environmental factors, not genetic factors, because of the time scale that we're seeing such a large increase. And so focusing on inflammatory bowel diseases, like I said, Crohn's disease is one of the major diseases encompassed by this. Ulcerative colitis is the other major disease. It affects millions of people worldwide. There's a rising incidence. Onset can be at a very young age or young adulthood. You have frequent relapses when you've got this disease. Frequently, you need hospitalization or surgery to treat. And in general, I think the quality of life of individuals that have IBD is greatly impaired. And so we want to understand what's happening in this disease. So when we think about it, we know that there's a genetic component, and so a lot of work has been done, much of it actually at the Broad Institute, looking at genetic risk variants that contribute to IBD. We also know there are environmental factors that can affect IBD, and really the interplay between the gut microbial community, the intestinal epithelial cells, and the immune system is what's changing in this disease. So we want to understand now, how do we go from looking at what bacteria are present to actually getting some function of what those bacteria are doing. And I'll ask three main questions that I'll come back to throughout the talk, where I'll give you some examples of what we're doing to address them. So the first question I kind of alluded to already is are bacterial strains different in disease versus healthy samples, even for the same species I'm talking about now? Are there genes that are differentially expressed in health or disease? And are there microbial metabolites that are altering host either immune cell or epithelial cell function in the gut? And so the first question we looked at using this particular study, the longitudinal stool study. So this is one of the first cohorts we had, where we had samples. Each row here is an individual that was tracked, either healthy in green or with IBD in the other colors. We got samples monthly from these individuals and performed whole metagenome sequencing on all of the samples. The cohort had patients that were adults as well as kids, and we used a validation cohort that had already been published to validate what we were observing. And so with these data, we were able to look at different species that were increased in inflammatory bowel disease or decreased in disease. Brantley Hall, who was the postdoc working on this project, was staring at this and realized that one of the main things that's different here is the aerotolerance of these microbes. So all of the species that were decreased in IBD are very strict obligate aerobes, whereas the microbes that are increased in IBD tended to be facultative anaerobes. So this means that the facultative anaerobe means that they're able to still grow in the presence of oxygen, whereas the obligate anaerobes die. And so the hypothesis that we reached, and this was based on our study as well as others that had been previously done, was that an increased production of reactive oxygen species during inflammation in IBD may be contributing to the decrease of the obligate anaerobes and the replacement by these facultative anaerobes that could tolerate the reactive oxygen species better. And so what Brantley did he manually curated a list of all of the species that were present in the samples, and was able to assign them either facultative anaerobe, strict anaerobe, et cetera. This is a non-trivial thing. It's actually very hard to compile all of those data to do that, because a lot of it has to be based on experimental work that's been done. But what he saw was striking when he assigned the classification of facultative anaerobe to the microbes. He saw that in inflammatory bowel disease, there was a much larger proportion of samples that had these facultative anaerobes. There's almost none in the control samples in our study. He looked at the Healthy Human Microbiome Project samples. Almost no facultative anaerobes were present in those. And in this validation cohort that I mentioned, again, he saw the same pattern where in inflammatory bowel disease, there were many more microbes that were facultative anaerobes. From the list that I showed you previously, you may have noticed there was one organism in our increased in IBD list that was not a facultative anaerobe based on published data. That was Ruminococcus navis. But despite the fact that this is not a facultative anaerobe, this organism showed the same pattern of being increased in abundance in IBD. And so the question we had then is Ruminococcus navis more aerotolerant than we thought? And so Ashley Gardner in the Xavier lab did a very simple experiment where she looked at Ruminococcus navis, Eubacterium elegans, which is a very strict anaerobe, and E. coli, which is a facultative anaerobe. And basically she took the cells, she grew them up in the anaerobic chamber, then she exposed them to oxygen for either 1 hour or 3 hours. And with the strict anaerobe, you see that once you expose them to oxygen, even after just 1 hour, we get almost no viable cells after that exposure. E. coli, we know, can grow both in the presence of oxygen or not. It grew fine even after the oxygen exposure, and Ruminococcus navis was somewhere in between. So here we saw that after even 1 hour of oxygen exposure, we lost very few microbes. So they weren't growing, but they also weren't dying. And even after 3 hours of exposure, we still had half of the population that was viable. So we think that Ruminococcus navis is, it's certainly an anaerobe, it's not facultative, but it can survive the oxygen presence much better than other strict anaerobes. And so we dug deeper on Ruminococcus navis to see if we could observe anything in the samples that we had. And so here I'm looking at just Ruminococcus navis abundance. Each row is an individual. Each column is a time point that they were sampled at. And these time points are colored by abundance of R navis. So what you can see here is that we're observing transient blooms. So in a given individual, you'll have very, very low abundance of navis, and then you'll have a bloom. And we saw this over and over again in different samples. We looked in the validation cohort that I mentioned. They had 4 time points there, and it was the same thing. So Ruminococcus navis was blooming in disease. If we had just a single time point, we would never detect this, and we actually hadn't detected it previously. If we had only 16S sequencing data, we wouldn't detect this because Ruminococcus navis is one of the microbes that is very hard to classify by 16S. So because we had metagenomic data, because we had longitudinal data, we were able to see that navis was peaking during disease. In some cases, we saw that peak corresponded with an increase in disease activity. So we wanted to dig deeper on R navis. We looked at all of the published genomes that were present. There were not that many of them. So we isolated more navis from stool samples from people that have IBD. We sequenced those genomes, and we could determine all of the gene clusters from all of those genomes. That's called the pan-genome. And then we could map our samples to those genes. And so in dark blue is the presence of those genes. You could see that some genes are present in all of the Ruminococcus navis samples. So our core genes that or our marker genes that I was mentioning that we use for classification would be some of these that are present in all samples. Some genes are present in in some samples, but not in others. And we saw that when we made a tree based on the presence or absence of these genes, we saw 2 clades that formed. And so we wanted to look at which samples were present in each of those clades. And so using all of the data that we had from the LSS cohort, the validation cohort, healthy cohorts, we observed that in clade 1, we always saw samples from both healthy individuals and individuals with IBD, but clade 2 only had samples that were from our IBD patients present in it. So we did not find any adult metagenomes that mapped in this clade. And so we compared the genes that were present in clade 2 versus clade 1, and there were 200 genes that appeared to be specific to this clade that was really enriched for IBD samples. Then we could go back and we could look, because we sequenced the genomes, we could look at what are these genes that are different. And you could see that not all of those genes are present in every single sample here, but none of the genes are present in the healthy samples. So at the top here in green, those are the healthy samples. None of these genes are present in them, and the genes are present in some of these samples. The genes were enriched in those that are responding to oxidative stress, those that are giving the microbe the ability to utilize mucus, genes for adherence you could see, as well as lots of genes involved in iron acquisition. So these are all factors actually that could contribute to how the microbe interacts with the host in the gut. Okay, so here I'm wrapping up that story. Basically, we've shown that there are different strains that are present in disease. We can even get at what genes are different in those strains, and now we can start to dig deeper into the functional relevance of those genes.
Can I ask a question?
Of course.
So do you know if these come with the disease, or are these already there? So what is the direction of causation? Does this correlate with the, say, a genetic risk for IBD?
So I don't think that we have enough data to answer that question, but I will say that we have some of those individuals, we saw them over time. And one individual in particular had all clade 1 in every sample. They had a flare of IBD, and at that sample, we saw that the clade 2 came in. So based on that, I wouldn't say that it comes with IBD per se, but rather I think there's something with the environment when you're having an active flare. Okay, so the second thing I want to talk about is looking at gene expression in microbiome studies. And here I'll talk about HMP2. So HMP1 was all healthy individuals. HMP2 is the second phase of the Human Microbiome Project, where we focused on disease. And the disease, like I said, that we were focusing on is inflammatory bowel disease. This is a longitudinal study. We sampled individuals biweekly for a year. We got lots of different data from these individuals, including host genome data, clinical data, genetics, transcriptomics, metabolomics, et cetera. But for the pilot study that I'm going to talk about, I'll focus on just the metagenomic shotgun sequencing data and metatranscriptomic sequencing data. So for the pilot cohort, we aimed for getting at least one sample from all of the subjects. So here are all the individuals with Crohn's disease, our healthy controls here, and ulcerative colitis here. We have long time courses for a couple of individuals in each of these groups, and we have several shorter time courses as well that are represented. Open circles means that we have metagenomic sequencing data, and a filled circle means that we have matched metatranscriptomic sequencing data. And so when we get these data, actually processing the metagenomic and the metatranscriptomic data, once the data are generated, is very similar. So we can do some quality control. We can get rid of bad sequences. We can get rid of host DNA that may be contaminating our whole metagenome sequencing. And then we could use MetaFlan to taxonomically profile the organisms in the samples. We can also do functional profiling of all of the genes present. In this case, we used HUMON2, which is another program available through the Huttenhauer lab. I'll give links to all of these later. But HUMON2 allows you to look at gene families that are present, metapsych pathways that are present, as well as GO categories. So you can start to look at the functions of the genes that are present in each sample. And then you can couple that with the metadata and clinical data that we have. So the first big overview here is just looking at DNA and RNA abundance for every species that we've seen. Now we're looking at pathways is what this plot is, but you get very similar results with other classifications. So looking at the pathways, you could see that in general, if you've got DNA for a given organism, you have similar abundance of RNA for that organism. The dots are colored based on the correlation. So if we look at one of the most highly correlated organisms, Parabacteroides meridae here, you can see that at the DNA level, so now each column is an individual, and the bars are showing pathway abundance for all of the pathways. So every color is just a different pathway that we're looking at. But you could see that if you've got DNA present for a sample for Parabacteroides meridae, you see that there's also RNA for the most part. You see that some individuals don't have any organisms that with reads mapping to this species, and those are blank here. But if we look at other organisms, so Dialyster and Visis, for example, we saw that there was relatively equal abundance of DNA for these two organisms. If we were looking at the metagenomic data, we'd think they were present in the same abundance. They were present in samples from people that have IBD or don't. But regardless of presence, there was almost no RNA for this organism. So even though this microbe was there, it's likely not doing anything. It may be dead. This is an oral microbe, or a microbe that's been characterized as being found in the mouth previously, and so it's possible that it's just passing through. Based on the DNA abundances, though, we would never have seen that it was doing nothing. Now we could look at individual pathways. So what I'm showing you now is a specific pathway. This one just happens to be this arginine ornithine de novo biosynthesis pathway. Now each column again is an individual sample, but the bars represent the overall relative abundance of the pathway, and they're colored based on which species are contributing the pathway. So from the sequencing information based on the reference genomes that we have, we can say that the reads are mapping to a given species. And so here, looking at the overall relative abundance of the pathway, you could see that at the DNA and RNA level, they look fairly similar. If we want to compare the species, though, it's a little bit easier to visualize if you look at the pathway relative abundance. So now each of those has been scaled to 1, and you can see the colors are representing which microbes have this pathway. So in some cases, at the DNA level, you could see that there's very little of a given microbe that's contributing, and at the RNA level, you see a huge amount of the reads that we have are from a different organism. And the same goes here. Sometimes they look very similar as far as the DNA and the RNA, which microbes are contributing, and sometimes they're different. So if we look at a single individual now for the same pathway, we could see that over time, this individual has 2 to 3 microbes that are contributing the pathway. They're colored in different colors here. But at the RNA level, it's basically taken over by only 1 microbe. So the microbes appear to be sharing their function. They can tell what other microbes are doing. In this case, they're probably secreting ornithine or arginine, I don't know, in this pathway, and they can sense that. And so at any time, only 1 species is expressing these genes. Interestingly, it was never the same, or it wasn't the same species at different time points. So they're kind of taking turns. Okay, so we know that there are genes that are expressed differently relative to the DNA presence. We know we've been looking at strains. I'm going to give you one last snippet where we're trying to understand now what are those microbes doing as far as the molecules that they make. So in this example, you need to think about what's happening in the gut. So we have epithelial cells that are present. The microbes are present in the lumen. There's an inner and an outer mucus layer that's produced by the host cells. The inner layer has no bacteria in it, and the outer layer is very rich in bacteria. The mucus is essentially a protein that's decorated with a lot of different sugars, and some bacteria can actually break those sugars off and consume them. So it's a really nice environment for some organisms. And there's molecules that are present there that are produced either by the bacteria, some are produced by the host, some are coming from food that we eat. There's a lot of different molecules present in the environment. So we know that in IBD, the mucus layer is thinner, and we hypothesized that identifying bacteria that can grow on mucin, so that can break down those sugars and consume them, might enable us to identify commensals that are important for gut homeostasis. And so we looked at, here's the mucin peptide, here are all the different sugars in different shapes shown here. We made a list of genes that were responsible for cleaving these sugars off of the mucin, and we also made a list of genes that were responsible for taking those sugars up. So they need to be transported inside the cell for them to be used. We then looked for those genes in healthy individuals. So we used the Human Microbiome Project 1 data set here. And we saw that bacteria that had these genes tended to come from Bacteroidetes or Clostridiales members. So these are the order of the organisms that were mainly able to do this function. So then we tested, so we had a bunch of isolates in the lab. We tested their ability to actually grow on mucin, and we saw that more than half of the microbes we predicted would be able to grow on mucin actually were able to grow on minimal mucin medium in the lab. And so then we wanted to know, are these mucus-utilizing species beneficial to the host? To make a very long story short, we did a mouse experiment where we gavaged mice with some of these bacteria that could grow on mucin. We induced epithelial cell injury using this DSS model, and we monitored weight loss. And what we saw is that the bacteria that were gavaged, that were able to utilize mucin, one of them, Peptostreptococcus brucelli, was very protective in this model. So the mice didn't lose weight, and it did even better actually than Akkermansia mucinophila, which was our positive control of an organism that's known to be able to grow on mucin that has beneficial properties. We also looked at expression of MUC2, which is the gene necessary for mucus production. And we saw that in those same mice, there was an increase in expression of MUC2 when the mice were treated with P. russellii. And so we wanted to know what could be mediating this protection, and if other Peptostreptococcus species were also beneficial. And again, to make a long story short, we looked at genomes. We genome scanned many different Peptostreptococcus species genomes. We honed in on this particular cluster, the phenolactate dehydratase gene cluster, that has genes that are involved in tryptophan metabolism. And these had been previously shown in another species to be responsible for making indole propionic acid, which was a beneficial molecule. So some of these species have the full cluster, and one of them, P. stomatis, did not have the full cluster, so we predicted it would not be able to make these molecules. And when we used mass spec to search for the molecules, we saw that indeed in culture, P. russellii and P. anaerobius were able to make these two tryptophan metabolites, indole propionic acid and indole acrylic acid, whereas P. stomatis was not. and a third metabolite that's a phenylalanine metabolite, which should not be affected by this cluster, was produced by all 3 strains. And so now we wanted to see what are these molecules doing. And we set up a system that's kind of a reductionist version of the gut. So here we've got epithelial cells in these organoids. These are differentiated cells where we've got stem cells and cells that can produce mucus, which are these goblet cells, as well as other epithelial cells. And we co-culture them with immune cells, which allow us to monitor an inflammation level. And so in this co-culture system, we can add our molecules that we're interested in, and we can measure, so that's what they actually look like. You could see the macrophages here and the organoid here with epithelial cells. And we can measure MUC2 expression again. So this is tells us if the mucus is being made. It's a goblet cell marker. You could see that when we treat the organoids with indole acrylic acid, we see an increase in MUC2 expression. We didn't observe the same thing with IPA or our controls. Then when we looked at cytokine production, the anti-inflammatory cytokine IL-10 was induced in the presence of indole acrylic acid as well. So this is really nice. This means that the molecule we're looking at protects against DSS-induced colitis. It causes the cells to make more mucus and stimulates the production of anti-inflammatory cytokines. And so we wanted to know if this was relevant in human cells. So we looked at human PBMCs, and we performed RNA-seq on cells that were treated with indole acrylic acid or DMSO. And there were a couple of categories of genes that were altered. So we saw a huge decrease in genes. These are now host genes involved in oxidative stress and inflammation. And we saw an increase in genes, many of them regulated by the NRF2 pathway that protect against oxidative damage. And so we saw this, we said, hey, let's look at a NRF2 reporter, so to directly see if there's a difference in expression of the reporter, and we saw that with indole acrylic acid there was a difference. Is there a question? Indole acrylic acid. And so now we wanted to look in disease. Could we see the genes that we now think are important for this this phenomenon in our metagenomic samples from people with disease? So we looked at a cohort of IBD patients. We had stool metagenomic data, and we looked for the genes involved in mucus utilization. So the ones that are responsible for cleaving the sugars off of mucus and transporting them. Now, once again, we're back in bacteria. What we saw is that IBD patients had a reduced capacity to cleave mucin. There were fewer of these fucosidases and sialidases that are able to cleave those sugars off. So the organisms that are necessary to eat the mucus are not as present in IBD. We also looked for the FLD gene cluster, which is the one responsible for producing the indole acrylic acid that I was talking about, and we saw that patients had less capacity to metabolize tryptophan into these molecules. There were fewer samples that were positive for this gene cluster in the individuals with ulcerative colitis or Crohn's disease. And so the big take-home points that I want to leave you with is that we're looking at the microbiome in lots of different ways. Individual microbiomes are unique and complex between individuals. There are differences in the strains of a given bacteria that are present, and these strains can actually have niche adaptation even within your mouth. Different parts of your mouth can have different strains of a given species. There are differences in the strains that we observe in disease, so as we start to look more at the strain level, I think we're going to see more of this, these differences come up, and we can start to now look at the functional potential, what genes are present in those strains versus not. In addition, by looking at the metatranscriptomic data, we can look at functional activity instead of just functional potential. And we're starting to identify bacterially derived molecules that target pathways important in the gut, such as barrier function in epithelial cells and cytokine production. And I just want to say there's a lot more to be learned. I think this field is really just at the start, and looking at what's going on in the microbiome in all of these different ways, I think will help us to better understand what this community is doing to contribute to health or disease. I did say that I would talk about all of the programs that I mentioned. So these are just the ones that are available through the Huttenhauer Labs BioBakery. You can just Google BioBakery and find it. All of them are here, along with source code, documentation, tutorials, etc. All of the work was done in the labs of Ramnik Xavier and Curtis Huttenhauer. I tried to put references as I was talking. The references are here, and I'm just highlighting some of the key players in the different stories, but many people contributed to all of these projects. And I'd be happy to take any questions.